All right, thanks a lot for attending the workshop today. Excited to share what we've been working on at Arima, and hopefully this is informative and insightful set of presentations. So what I'll be talking about is 3D genomics, how a 3D genome approach to oncology reveals novel disease mechanisms and therapeutic targets. I assume that several of you may not know who Arima is, so I'll introduce ourselves, talk a little bit about what we mean by 3D genomics, assuming that you haven't heard of that term before, and then how you can leverage 3D genomics to detect structural variants. And I'm going to use the term fusions just as sort of a catch-all for all different types of structural variants, translocations, inversions, etc., that fuse two pieces of DNA that shouldn't be fused together. And then in the second half, how we've applied 3D genomics technology in collaboration with our clinical partners, such as Sarah, to identify targetable diagnostic and prognostic fusion biomarkers in so-called driver-negative tumors. So these are tumors that have been previously profiled by clinical DNA or RNA sequencing and don't have a targetable or diagnostic marker. So a little bit about Arima. We are advancing the life sciences through the power of 3D genomics. And in doing so, driving biological discovery in genomics. And in certain contexts, we believe this technology can improve cancer diagnosis and optimize patient care. You'll see some of that evidence today for myself as well as Sarah. So what is 3D genomics? I like to introduce the technology to compare and contrast it against what I call linear genomics, which is really just the conventional DNA or RNA sequencing, where lots of us, when we think of the genome, we think of it as this sort of linear entity, maybe looking at a genome browser or looking at genes. It's always shown in sort of a linear fashion, A's, T's, C's, and G's. But in fact, as many of you all know, if you actually look into cells, the DNA is organized into 3 dimensions. And what the 3D genomics technology is it's really the marriage of sequencing and structural information. So I always tell people it's sort of like sequencing the structure of a chromosome. Kind of like fish is like imaging the structure of a chromosome or the spatial positioning of genes, or sequencing it and measuring the spatial positioning of genes, just like fish, but using NGS as sort of our means of how we do that. You'll see that on the next slide. So this is sort of just the one workflow slide here, and just sort of how the secret sauce sort of works is our technology, which is called Arima Hi-C, is really just a glorified DNA extraction technique, where you start with a sample, and this could be an FFPE sample, blood, frozen tissue, etc. The first step is to cross-link the cells, and if you have FFPE tissue, that work has already been done for you. You don't have to cross-link anything. And there's really 2 core steps for how we capture the 3-dimensional information of the genome. And those 2 steps are right here. So if you have 2 pieces of DNA that are held together in close spatial proximity, because that's how the DNA is organized in cells, this purple piece here and this blue piece here could be from very far apart along the linear genome, but they've folded together in 3D space, just on sort of native organization of DNA. And there's 2 little tricks that we do. We cut the DNA, and then we ligate it back together. So two pieces of DNA that could be very far apart along the linear genome, but were held together in 3D space, have been cut and then joined together. And in doing so, that cutting and ligating preserves information in that DNA molecule that those 2 pieces of DNA were spatially proximal to each other within inside that cell. So now all we really want to do is sequence this piece of DNA. And that is exactly what we do. So once we've extracted the DNA that has been through the process of digestion and religation, that can go into a standard Illumina sort of library prep, plus or minus target enrichment, Illumina sequencing, and then on the back end, the informatics to identify the fusions that I'll be talking about today. So, that's sort of at the molecular level what's going on, but conceptually, how do you use this to identify fusions? So first, consider a karyotypically normal cell. You have two different chromosomes shown here, chromosome A and chromosome B. First, I'm going to show you kind of the conventional paradigm for identifying fusions. Well, in a normal cell, you sequence chromosome A, chromosome B, there's no fusion, you don't find any fusion reads, and then in a cell with a fusion, where chromosome A and B have, let's say, fused together, then the evidence of conventional sequencing approaches are basically the reads that are part gene A, part gene B, and that's the sort of chimeric reads that support the fusion call. With the 3D genomics approach, what we know is if you look at cells, let's say this is chromosome 9, let's say this is chromosome 22, well, in karyotypically normal cells, those two chromosomes, they're not just randomly intermingled within the nucleus, they actually have their own, and they sort of occupy their own three-dimensional space, and they don't kind of crosstalk to each other. And so what our technology is measuring is these contact points between DNA. We call them 3D linkages. And so here on chromosome A, our technology would cross-link and ligate those two pieces of DNA and then fold it onto each other here, here, and then within chromosome B, these three touch points. And if you project this sort of ball of spaghetti onto sort of a linear space, these cross-linked and ligated reads are going to be represented like this. This piece has been cross-linked and ligated to this piece along chromosome A, and so forth here on chromosome B. And this is in a karyotypically healthy cell. But then imagine a cell with a gene fusion where chromosome B and A are now fused together as one chromosomal entity within that cell. And now there's many different touch points between chromosome B and A, shown here by those green dots, which you do not detect the crosstalk between those two chromosomes in a karyotypically normal cell. And if I show this sort of as a linear representation, what you can see is you have these reads that are derived from the gene A area that are cross-linked and ligated across the breakpoint over here to the gene B area, which those do not happen in a karyotypically normal cell. And this is just an example of one cell, but of course you're going to profile tens, hundreds, or thousands of cells in a single FFPE slice, let's say. And so this is cell number 1. There'll be another set of interaction points along the DNA in cell number 2. And then those linkages continue to pile up, those reads continue to pile up from gene A to gene B across cell number 3 until you've analyzed tens, hundreds, or thousands of cells, and you see this massive amplification of the fusion signal by aggregating sort of all of this signal in the neighborhood of A linked to the neighborhood of B, and that's the evidence that we try to harvest to identify gene fusion. So we sort of think of it as signal amplification. So the main takeaway here is the conventional approach, you're looking at just the reads at the breakpoint, and with the 3D genomics approach, you will have some reads at the breakpoint if you scan your eye down across here, but on top of that, you'll have all the linkages that hop over the breakpoint. I know this is just a cartoon, so here's one real-world data example where we had a CML patient with a BCR-ABL fusion, and if you just look right at the breakpoint between BCR and ABL, well, our data has 78 reads that are chimeric at the breakpoint, and if you run the same assay in a healthy sample, there's obviously 0. Now let's take a step back. Now let's look at the whole broader BCR gene and the ABL locus and see if we can see the linkages that happen between those two genes in addition to the ones that are there at the breakpoint. So what I'm showing here is that zoom out view. That green box that you can see is now much more narrow because we've zoomed out. That's looking right there at the breakpoint. And I think what's fascinating about this image is the way that we made it was, this was with an exon capture panel where we captured the exons of BCR. So you see kind of a pileup of these reads at the exons of BCR. But what's so cool about this image is every single read that's piled up on an exon over here is spatially linked to another read in ABL1 over on this side. So it turns out that there's 78 reads at the breakpoint, but if you count all those linkages between BCR and ABL1 in totality, it's 7,200 reads, or 100x signal amplification of that particular fusion. And when you run this in a karyotypically normal sample, well, in this case, there was 0 reads between BCR and ABL, kind of like the background signal that we would compare against. The other way that these data are represented, and Sarah's going to represent her data this way, so I figured I'd show a couple slides about this, but not go too deep into the weeds, is because our technology is really measuring the crosstalk between 2 pieces of DNA, it's frequent to show the data as this type of heat map, where each chromosome is a column, and each chromosome is also a row. And the red signal is the number of reads between all pairs of loci genome-wide. And in a karyotypically normal sample, what you see is there's lots of those 3D linkages that happen within chromosomes, but very few. It's almost a complete white space when you look at crosstalk between 2 different chromosomes. So this box right here would be the crosstalk between chromosome 1 and chromosome 2, which doesn't happen in a karyotypically normal sample. But if I show you a formalin-fixed paraffin-embedded colorectal cancer tumor with a ROS1 SLC34A2 fusion, which is between chromosome 4 and 6, you get this exquisitely strong signal between 2 different chromosomes because, in fact, they've actually joined together and they are one chromosomal entity within inside the nucleus. And so you get this sort of scorching red signal, which our algorithms use to find those fusions. Right, so these sort of evidences here in the off-diagonal space would be interchromosomal translocations. You can zoom in on this. This is a very broad view, but you can zoom in kind of at the gene level. So let me zoom into the ROS1 fusion between chromosome 4 and 6. And I don't have time to go sort of into deep detail of like how to interpret this plot, but ultimately this purple line right here is the breakpoint on chromosome 4, and it bifurcates the SLC34A2 gene. And then this purple line is the breakpoint on chromosome 6, and it bifurcates the ROS1 gene here. And so the 5' end of SLC34A2 is fused to the 3' end of ROS1. So that's how we interpret the data. And just one thing to sort of introduce the technology is we've run concordant studies with several different clinical partners where we compare our technology to identify known gene fusions that were found with orthogonal methods like Archer panels and FISH and so forth. And in every case where there is a fusion, we've been also able to find that fusion, so we are concordant with these orthogonal methods. And also robust across a whole variety of different tumor types, which are shown over here on the right. Okay, so in the second half, I want to start with a very interesting story that kind of actually set us down this path of exploring the viability of our technology in precision oncology. It all started back in January 2021, where there was a stage 2 glioma that presented and was resected. This was in a 15-year-old girl. That biopsy was profiled by NYU's Genome Pack, which is a 607 gene FDA-cleared panel, as well as their Archer panel. We weren't able to find a driver alteration with those clinical NGS panels. The patient was put on chemotherapy and was monitored, and then 6 months later recurred with rapid progression. And we ran the same 607 gene panel, the same Archer panel, still didn't identify a targetable mutation. Then I had been collaborating with a pathologist there, Dr. Schmitt, on a research paper finding fusions in pediatric brain tumors. He sent the sample over to our lab in a research capacity. We profiled it, identified what we thought was a novel PD-L1 fusion. And this, I told Dr. Schmitt, he ran a confirmatory IHC test in his CLIA laboratory, came back diffusely positive. The patient was given pembrolizumab in September of 2021. 1 year later, at the 1 year follow-up, still had stable disease with no new tumor growth. And so, while this was a great story, we were also wondering, well, is this sort of an N of 1 case where we just got lucky? So we kind of asked, well, how many other patients are there like this with advanced cancer who don't have a druggable target? And based on which technology is used and who you ask, or the different tumor types, I think the numbers vary, but approximately 50% we think of those advanced cancers don't have a druggable target. So we were thinking that I guess there's an opportunity here to make quite a big impact, at least in that population, if the numbers hold up at scale. And so we designed a series of studies to try to ask, well, in these driver-negative patients that don't have a druggable target after clinical DNA and RNA sequencing, how many of them are like the 15-year-old girl where we can find something that could be potentially actionable? And we did this in a retrospective setting, so we don't have outcome data, but I wanted to summarize those results. So what we found is, of those 221, 70 of them have a fusion that was identified with our technology, where the gene in or around the fusion breakpoint is the target of an FDA-approved therapy today. So that's about a third. 12 of those patients had a fusion where the gene linked to that fusion was the target of a drug in an ongoing clinical trial, and 29 of them, the gene associated with the fusion was either diagnostic or prognostic in that tumor type. So if you sum all those up, about 50% of those we think could be clinically actionable. And instead of looking at the patient level sort of waterfall, if you look at individual tumor types, as shown here on the left, this is the percentage of this sort of tier 1, tier 2, tier 3 in each tumor type. And a lot of the data is coming from central nervous system tumors, primarily pediatric. Also have 23 GIs, 24 solid hemes, lymphomas and plasmacytomas, and then a cohort in collaboration with Sarah on gynecological sarcomas, mostly uterine sarcomas. So she'll be speaking to that in a bit. Here's just sort of a smattering of the different biomarkers I identified, and I'm going to show some examples of these in just a minute. But I think what we think is super interesting part of this data is when you ask what types of fusions are we finding. And one of the buckets, which is sort of obvious, is the typical gene-to-gene fusion that I think everybody thinks about when they think of the term fusion, like BCR-enabled. And we find typical gene-to-gene fusions involving the NTRK genes, estrogen receptor, NRG1, breaks in RAD51, which Sarah is going to mention, FGFR, etc. But there's another type of fusion, which I think is well known and established, at least in the heme community, less so appreciated in solid cancers, I think, where you have rearrangements with non-coding regulatory elements. So, classic example here in heme is IgH rearrangements with cyclin D1, but the main point here is that the actual breakpoint where these fuse together don't produce a fusion transcript, and so they're invisible to RNA, which is looking for fusion transcripts to identify fusions. So these are typically detected by FISH. And a bulk of what we have found of those tier 1, tier 2, tier 3 level biomarkers are these sorts of events. Not all of them, but about 75% of them are, including genes that you would recognize like NTRK1, but also diagnostic or prognostic markers like platinum. Also, here is the PD-L1 that I showed you in the very beginning. And if you think, I kind of already alluded to this, that RNA is not going to be able to see these because the breakpoints outside of the gene body, they don't create a fusion transcript. But how is our technology able to find these when they're outside of the gene body? Well, envision a scenario where you've designed probes to pull down gene B and whatever gene B is spatially linked to. If the breakpoint is outside of the gene body of gene B, let's say it's right here, well, gene B is going to be cross-linked and ligated to its sort of local neighbors. And when you have a fusion, those local neighbors are going to be derived from a different chromosome. So we're still able to sort of see these fusions even when they're outside of the targeted gene. So we're aggregating all of this signal, still able to find the fusions. But in a conventional approach, if you're targeting gene B, these are going to be invisible. Just a couple examples here. This was very fresh new data as of this past week. So this is kind of an interesting case of a diffuse large B-cell lymphoma with one of these promoter swapping events where the breakpoint was actually right here at the promoter of BCL6, and its fusion partner right here is the IGH locus of chromosome 14. But what was really interesting about this case is, and it was BCL6 positive on IHC, but this same sample also had a PDL1 fusion rearranged with another allele on the IGH locus, which you're seeing here. And this just came, I think, yesterday, this is about as positive as it gets for PDL1 expression, diffuse on the lymphoma cells, negative on the blood vessel cells, which you can't really see. So this is in heme, and I think this sort of mechanism is generally, I think, established and accepted in heme. But how about solid tumors? This is a SEG brain tumor where the NTRK1 gene is not at the breakpoint. It's actually 66 kb away from the breakpoint. But we would hypothesize that perhaps this is an activating event, similar to how those rearrangement events are activated in heme. It turns out this is a IHC positive by NTRK, even though it doesn't have a typical NTRK gene to gene fusion. It has a breakpoint that's adjacent to the NTRK gene. And we think that this is leading to activation in the absence of a typical gene to gene fusion. We tried to do as much validation as we can. Some of it's limited by what types of antibodies are available. I'm not going to go through all these in detail. We can talk about that afterwards. And lastly, we've been trying to, I think, analyze as many prospective patients as possible, obviously on a research basis where the findings have to be confirmed with a CLIA validated test. And we have 6 of those so far, 2 PDL1 rearrangements, one in a glioma, one in a TNBC. Both got pembro. Both have responded very well. An NTRK case, MIPL1 fusion that spared the patient unnecessary adjuvant therapy, and a couple others where the patients were too fragile for therapy. I'll just leave by just sort of an update on a couple of the partnerships in the clinical space. We recently partnered with Perion, now Valsera, to provide streamlined and accurate NGS data analysis, interpretation, and reporting for our planned clinical offering. And we've also partnered with Protein Biodiagnostics in Florida to make our technology available in their CLIA CAP laboratory this year. Just a plug, very interested in research collaborations like the ones that we've done in the past that you hear about with Sarah. In these 4 disease types, we were interested in profiling samples to look at the prevalence of gene-to-gene fusions as well as proximity fusions in a consecutive case series. So if you have or know anybody who could be interested in that, please contact me. I'd like to thank all of our partners. Really, it was Darren Siegel, Rajiv, Nidhat, and Atif who were the very first believers in the technology, and then Mattia has been really the tip of the spear on the driver negative sample analysis in collaboration with Sarah at MSK. Thank you all for your attention. Hopefully that was insightful. We'll pass it to Sarah.
All right, well, thank you very much, Anthony, for inviting me to join you in this talk today and in this forum, and also thank you all in the audience for coming, particularly when there are so many exciting events at this conference this year and also a lot of fun attractions in the city. So I wanted to give you kind of a clinical perspective of the utility of Hi-C when it comes to pathologist workflow and the utility of this technology in gynecologic sarcomas, which is my area of interest. So, the classification of uterine mesenchymal tumors has really exploded in the last couple of years, mainly due to the increased utility of next-generation sequencing methods. So I just wanted to cover a couple of things that we use at MSK, and obviously there are similar assays at other institutions and at private laboratories. But pathologists have access to our our solid tumor FISH panels, which covers 47 genes that are commonly rearranged in solid cancers. And then there's MSK Impact, which is similar to the NYU Genome Pact. It's our targeted DNA-based NGS assay that looks at 505 genes. And within that assay, there is a very small fusion panel that covers 70 introns of 20 commonly rearranged genes in solid cancers. So for FISH and targeted DNA sequencing, you use those to detect known fusions. We have a targeted RNA-seq panel, which is based on the Archer multiplex PCR technology. It's customizable, and so currently we are now covering 123 genes. The advantage of this is that not only can you detect known fusions, but you can uncover novel fusions. And that's where a lot of the discovery of new uterine sarcoma subtypes has really, this has really aided that. In addition to NGS, of course, many of us are pathologists here, but the importance of it is actually the coupling of what we see under the microscope and what NGS detects. So it's that genotype-phenotype correlation that has really led to our recognition of new uterine mesenchymal tumor subtypes. So for uterine mesenchymal tumors, I think the 2001 study that identified JAZ-F's 12 gene fusions in low-grade endometrial stromal sarcomas from the group at Brigham and Women's really kind of started this. And then as you go down the timeline, really after 2012, we've been seeing these new entities like high-grade endometrial stromal sarcomas that have either YWHAE or B-core fusions or B-core and tandem duplication, followed by the discovery of NTRK fusion-positive fibrosarcoma from our group, as well as PGR fusion-positive epithelioid leiomyosarcoma, REV1 fusions, and a subset of uterine tumors resembling ovarian sex core tumors. And the ones that I've highlighted in red are particularly the tumor types that are really associated with a specific fusion or genotype. The other side of NGS being beneficial to us in pathology, besides being a diagnostic aid, is really, we can really aid in therapy, particularly for uterine sarcoma patients. And these are just some examples from our group, showing that NGS has identified low-grade endometrial stromal sarcoma-associated fusions in certain morphologically unusual uterine sarcomas that also harbor somatic TSC2 mutations. And so these patients with NGS are able to benefit from aromatase inhibition, as you do in typical low-grade ESS patients, but also they benefit from mTOR inhibition. The other subset of uterine sarcomas in which we found NGS to be particularly helpful in both diagnostic and therapeutic aspects is ESR1 hotspot mutations in endometrial stromal sarcomas. So, usually our targeted DNA sequencing panel is ordered by the clinician when there is unusual behavior in the clinical presentation of the tumor. So in cases where there are shorter recurrence intervals or more aggressive clinical behavior, the clinician would order targeted DNA sequencing on uterine sarcoma at our institution. And in this case, we were able to couple both the morphologic features in which there is high-grade morphology in an endometrial stromal sarcoma, or there is altered ER expression by immunohistochemistry, couple that with the presence of frequent ESR1 hotspot mutations. So these patients actually benefit from fulvestrant, and NGS sequencing has really aided in us being able to tease out the patients who would benefit from that. And finally, NTRK fusions. Of course, NTRK fusions are present in a wide variety of common cancer types, but a minority of those common cancer types. But they're disease defining for some rare cancer types, including uterine fibrosarcomas. And people at UCSF were able to show that one patient with widespread NTRK fusion positive uterine fibrosarcoma actually benefited from entrectinib. So really, NGS has really, not only from a diagnostic perspective, but opened avenues for new therapies for these uterine sarcoma patients. But, there is a big but. So, it's great what we're doing, but actually I would invite you to see our current GYN fellow, Doug Allison's poster tomorrow, Wednesday afternoon, where we talk about, cost effectiveness of targeted RNA sequencing in the diagnosis of uterine mesenchymal tumors. But I wanted to show you this diagram from that poster. The other spin on this is that we did a retrospective analysis of 54 uterine mesenchymal tumors that were really difficult to classify. They were seen from 2016 to 2019 and were mainly uterine sarcomas where an extensive immunohistochemical panel was done and we still couldn't fit it into a particular tumor category. These were all sent for our targeted RNA sequencing panel, and after extensive IHC and targeted RNA-seq, 43% of those mesenchymal tumors remained unclassifiable. 80% had no disease-defining fusion. So that's a large swath of uterine sarcomas that we still can't properly diagnose. So that's the diagnostic aspect. What about therapy? Well, it's great that we're being able to tease out these small categories of specific uterine sarcoma subtypes, but actually, it's still a problem from the therapeutic perspective on the vast majority of uterine sarcoma patients. So for example, these are NCCN guidelines for various types of uterine sarcomas. And for stage 1, completely resected disease, regardless of the histotype, the treatment is observation. And really, if it's a higher stage disease or the patient recurs, the treatment, regardless of histotype, is roughly similar. So there lies the problem with, currently we have all these molecular toys, including really large NGS panels, but they fail to identify drivers and therapeutic targets in many uterine sarcomas. And part of that problem is because the NGS panels focus on the coding regions of the genome. And, it's very difficult as pathologists to get high quality frozen tissue for research, especially in the setting of sarcomas. And then also, targeted RNA sequencing is really optimal only on tissue, formalin fixed tissue that's less than 5 years old. So if you've got, a block that is older than 5 years old, the RNA quality is really suboptimal for RNA-seq. So with that, we have been able to partner with pathologists at NYU, Langone Health, as well as Arima Genomics to really utilize this novel high-C technology to identify novel drivers and potential therapeutic targets in patients with uterine sarcomas. So over the next couple of minutes, I just wanted to present some case illustrations of where I think high-C has been very useful for us from a diagnostic perspective and also potential therapeutic strategies. So this case is a uterine hysterectomy specimen from a 50-year-old woman with high-grade endometrial stromal sarcoma. She had high-stage disease but was completely resected, and she received docetaxel and gemcitabine, as well as radiation, followed by letrozole. She developed a pelvic sidewall recurrence 12 years later that was surgically resected, and this is what the morphologic features are in the primary tumor seen on the left. On low power, it kind of looks like a low-grade endometrial stromal sarcoma, but on high power, it looks very epithelioid, which is very unusual for what we routinely see in any either low or high-grade endometrial stromal sarcoma. This particular case was positive for CD10 and also had some HND45. So the recurrence, which is on the right, just showed more extensive epithelioid histology. And so the diagnosis was really difficult in this particular case. That we were thinking about low-grade endometrial stromal sarcoma, high-grade endometrial stromal sarcoma, and even PEComa. And what was interesting is that, we tried to profile the primary tumor, which was from 2008, using RNA-seq, and that failed. And then we profiled the 2020 recurrent tumor, and that was negative for gene fusions. We were, at that time, we had also seen other tumors of similar histology that showed JASF, CIS12 fusion, and TSC mutation. So we thought, okay, maybe there is a cryptic rearrangement, and we would use FISH to see if there was a JASF rearrangement. That was also negative, and this patient had a primary resection and the resection of the recurrence in an outside hospital. And so the outside hospital had sent tissue for Kettering NGS, and that was where the TSC mutations were picked up. We also subjected the tumor to methylation profiling by using the Illumina Epic array, and the sarcoma classifier actually classified this as a low-grade ESS with very high confidence. And if you look on the right with the heat map, the cases in the green are high-grade endometrial stromal sarcomas with B-core and YWHAE abnormalities, and the cases on the right in the tan are low-grade endometrial stromal sarcomas, and then the cases in the green are TSC mutant uterine sarcomas. This current case, that patient sample is the one in black, and you can see that it clusters more with low-grade endometrial stromal sarcoma. Well, I sent this case to Anthony, and lo and behold, we were able to identify an EPC1 EED fusion, which is known in rare endometrial stromal sarcomas that targeted RNA sequencing failed to pick up. So this was actually very helpful from a diagnostic standpoint in confirming that this is indeed a low-grade endometrial stromal sarcoma that harbored a TSC mutation. And then it also illustrates the utility of Hi-C when you have poor quality samples or old samples that wouldn't be successfully sequenced by RNA-seq. So this was actually a very informative case for us. Case 2, this is a 72-year-old woman with a malignant myxoid mesenchymal tumor in which myxoid leiomyosarcoma was favored. She underwent THBSO. This is the morphology of the tumor. It's positive diffusely for Desmond and negative for SMA, and it showed strong and diffuse PLAG1 expression by IHC. So our group recently showed that a subset of myxoid leiomyosarcomas harbor PLAG1 gene rearrangements. And this is not unique to myxoid LMS. This is also found in other tumor types and other organ systems that show this myxoid chondroid matrix. So pleomorphic adenomas of the head and neck will also harbor PLAG1 rearrangements. And in our study, we showed that, actually a subset of patients with myxoid uterine sarcomas will have diffuse PLAG1 positivity, but they'll lack the fusion. So if you can see here at the table, the ones that are starred, those are the cases that were PLAG1 IHC positive, but targeted RNA-seq was negative and FISH was negative for PLAG rearrangement. And this observation has been made in other tumors that often have PLAG1 rearrangements, that there is a subset in which PLAG rearrangements are not found, and yet there is PLAG1 overexpression. Hi-C was able to identify a novel PLAG1 rearrangement in this case, leading to PLAG1 overexpression. So it really, we're able to identify that this is a PLAG fusion positive myxoid leiomyosarcoma with the use of this technology. Case 3, so this was a 68-year-old woman who had an endometrial biopsy. It was sent to us as a consultation, and it was difficult to classify based on its morphologic features and non-specific immunoprofile. And it was signed out as highly atypical spindled and epithelioid neoplasm with myxoid features consistent with sarcoma. So this tumor is positive for CD10, ER-PR, showed a little bit of myogenic differentiation with Desmond focal positivity. There was focal B-core and focal PLAG1, but it was negative for S100. TRK-A was positive, as well as pan-TRK, which covers TRK-A, B, and C expression, interestingly, and ALK was negative. Because of the TRK-A and pan-TRK positivity, we sent this case for FISH, NTRK1 break apart FISH, and that was actually negative for an NTRK1 rearrangement. In this particular case, Hi-C actually identified an NTRK1 proximity fusion that resulted in TRK-A and pan-TRK expression. And this is informative because NTRK1 FISH can lead to a lot of false negative if it's an intrachromosomal event, and you may get insufficient splitting of the signals by FISH. So that may be the reason why this was a false negative FISH result, but Hi-C was actually very informative here. And this patient did not come to MSK for treatment, but it raises the possibility, should she recur, or if she is high stage upon resection, that she could benefit from NTRK inhibition. Case 4, so this is kind of going on, not looking at diagnostic, but rather looking at the therapeutic potential of using Hi-C technology in the realm of uterine sarcomas. So this is a 76-year-old woman who has a stage 1B high-grade adenosarcoma with sarcomatous overgrowth, who underwent TLH-BSO, and she's has no evidence of disease 2 years after the primary resection. This was actually subjected to MSK impact, which showed a high number of copy number changes suggestive of multiple chromosome level losses and gains. HiC was able to identify an ATM fusion in this case. So the other interesting finding from this is that there are numerous foci of shattering or chromothriptic-like events in this particular case. So there were over 300 total breaks. Another case, similar findings, 58-year-old woman with stage 1A high-grade Müllerian adenosarc with sarcomatous overgrowth. Again, she was alive and well 7 years after the primary resection. This is what it looks like. And in this case, HiC was able to detect RAD51B fusion, again showing similar multiple foci of shattering or chromothriptic-like events in this particular case. So given those findings, we also, looked at a variety of types of uterine sarcomas. So we retrospectively took 48 uterine sarcomas. 26 of them were profiled by targeted RNA sequencing at MSK, and 25 of them were negative. The only thing that we picked up was a B-core ITD in a single high-grade endometrial stromal sarcoma. 5 of them were also subjected to MSK impact, with only one case that showed a missense ATM mutation. And you can see the variety of different histotypes of these uterine sarcomas in the table on the right. And HiC identified a large frequency of homologous recombination repair gene alterations in a variety of uterine sarcomas. And you can see a number of different genes involved in homologous recombination repair had structural variants with enrichment for RAD51B altered cases. So we were able to find that 20 samples, or 42% of our uterine sarcoma cohort, had HRR alterations that were not detectable by targeted DNA and RNA-seq. And that has really important therapeutic implications, particularly with the use of PARP inhibitors, and it expands the potential for using PARP inhibitors outside leiomyosarcomas. So I hope, with these case illustrations, I was able to show you the potential utility of HiC in identifying genetic drivers that are not detectable by DNA and or RNA sequencing. It's able to identify novel drivers, as you can see in some of these cases, but of course, a number of them require additional validation, particularly in confirming HRD. And it shows the utility of HiC in the setting of failed RNA NGS or poor quality DNA NGS. The other side of this is the potential therapeutic targets that might be uncovered by this technology, particularly the NTRK1 proximity fusion case, and also alternative mechanisms for HRD for uterine sarcoma patients. So I'd like to thank all the collaborators at Arima Genomics and NYU for their important work that they've done in this project. Also, members of the MSK GYN disease management team, as well as a number of pathologists who contributed the cases from NYU, MSK, and also King Edward Memorial Hospital in Australia. So thank you very much. And happy to take questions along with Anthony for the next couple of minutes.